Good day, everyone! We are going to report about the microtubules. First, the introduction. The microtubules were discovered by D. Robertes and Franchi in 1953 in the axon of medullary nerve fibers and were named neurotubules. The microtubules was first named as neurotubules because they modulate the axon fibers. The term microtubules was given by Slatterbach in 1963. The microtubules are electron microscopic structure found only in eukaryotic cellular structures, but absent in mature mammalian electrocytes, slim molds, and prokaryotes. The microtubules are absent in prokaryote organism. The microtubules, or MTS, are the largest element of the cytoskeleton. As we all know, the microtubules are the part of cytoskeleton. They are what we call cytoplasmic microtubules. The cytoplasmic microtubules are the one who maintain axon, maintain the shape of the cell, orient cellulose microfibers in plants, mitotic and meiotic spindle fibers for chromosome movements, and lastly, vesicle movement. The cytoplasmic microtubule are the one who are responsible in mitotic and meiotic spindle fibers, chromosome movement, and cell division. As we all know, we have two types of cell division, the meiosis and the mitosis. Let's proceed in axonal microtubules. The axonal microtubules are highly organized, stable microtubules in specific movement associated in subcellular structure. The axonal microtubules are the one who associate in the movement of cellular structures such as cilia, flagella, and attachment basal bodies. The central shelf or axoneme consists of highly ordered bundle of axoneme MTS. The central shelf or axoneme has composed of axonal microtubules. There are different organization and different stages in microtubules. First, the microtubules in interface. In microtubule interface, they are organized into radial pattern centered of the chromosome. Microtubules in metaphase is organized in a bipolar architecture. Microtubule has the responsibility in different kinds of stages and meiosis and mitosis. General function of microtubules. There are the various function of microtubules. Cell use microtubules to provide structural support. Cells also rely on the dynamic assembly and deassembly microtubules to allow them quickly reorganize the microtubules. The microtubules are the one who support the entire cell. They has the function to in dynamic assembly and disassembly of microtubules to quickly reorganize the microtubules. Cells can make their microtubule more or less dynamic. Different cells can have unique organization of microtubules and suit specific needs. Elongate cell process like axon, axopad, heliozoan, protest. Microtubules oriented parallel to each other, an axon or axopad long axis helps move things. The microtubules help the cell process and help the cell move. In developing embryo, extended growing central nervous system axon to peripheral nervous system inhibit an outgrowth stop progressive collapse back to rounded cell's body. The microtubules has the responsibility in embryo development. Found as core of axopodial process of heliozoan protozoa. Many microtubules arranged in spiral in individual microtubules transfer entire length of process. Plants play similar role in plants, affect shape directly by influence cell wall formation, found in cortex just below membrane during interphase, forming a distinct cortical zone. The microtubule has a responsibility in plants. They play a major role in plants in structural support and the formation of cell wall. So also have role in maintenance of cell internal organization, like for example organelle replacement. So this prop in microtubules CO in O, Google disperses to cell periphery, goes back to cell when inhibitors remove. Microtubules and organelles around cell. So move micromolecules and organelles around cell in the correct manner. Intercellular motility. So in the movement of micromolecules, they are half physical transport between compartments of this prop microtubules to transport dependent on them. So the microtubules are the one who transport the half vesicle in different kinds of compartments in the cell. So proteins made in neuron cell body move down axon and vesicle. So what I said is the microtubules has the responsibility to move the cell in vesicle movement. So different materials move at different rates. Faster rate is 5 nanometer per second, 400 day vesicles since attached to microtubules. So in microtubules, there are vesicles attached. Structures and materials moving toward neurons terminal are said to move retrograde. So in the structure and materials moving, the movement is interrogate. Other structures like endocytic vesicle that are formed at neuron terminals and carry regulatory factors from target cells move from synapse to cell body in retrograde direction. Example, axon filled with microtubules, MFS and IFS, evidence suggested that both antegrade and retrograde movement are mediated mostly by microtubules. Video microscopy shows vesicle movement along MTS. So, confirmed by AM of axons, molecular motors move vesicles along the MTS that serve as tracks. So, elements of cilia and flagella. So the microtubule is the multi-elements of cilia and flagella. Active components of mitotic meiotic machinery move chromosomes. So the microtubules has the responsibility in cell division to move the chromosome in the center of the cell and remove the spindle fiber. So in microtubules there are polymers of alpha and beta tubulin. The alpha and beta tubulin is the structure of the microtubules. Later, I will go into discuss that. Microtubules are hollow polymers of tubulin and heterodimers. 13 linear chains of subunits called protofilaments associate laterally to form the microtubules. Large bands between protofilaments are called stabilized microtubules, limits the subunits addition and subtraction to microheads. So, microtubules are polarized polymers. The plus end is crowned by B-tubulin and assembled faster. The minus end is crowned by a tubulin, alpha tubulin, and assembled slower. So, the plus end is what we call the beta tubulin, while the minus end are what we call the alpha tubulin. So, purified tubulin subunits assembled in microtubules. So, microtubules polymerization begins with the formation of small numbers of nuclei. Microtubules polymerize by addition of tubulin subunits to both ends of polymer. A critical concentration of tubulin subunits always in solution. The concentration for assembly to occur. So microtubules has assembly and disassembly proceed by unique process termed dynamic instability. So the microtubules constantly switch between phase of growth and shortening. The transition from growing to certain states called catastrophe. So the transition from shortening to growing is called the rescue. So a population microtubules grows and shortens asynchronously. Thus, structures of growing and shortening microtubules ends different. So first, the growing ends have extensions of protofilaments, while the shortening ends have curling protofilaments that bend back away. So let's proceed in microtubule structure. So the microtubules structure are composed of alpha tubulin and beta tubulin. When the alpha tubulin and beta tubulin combine to each other, it will call the dimer. So, microtubule are hollow cylindrical structure. They are observed under electron microscope. Outer diameter is 25 nanometer wall. Thickness is 5 nanometer length along the breadth of the cell. Microtubules are made up of globular subunits. The globular subunit consists of single protein molecule tubule, what we call tubulin. So the globular subunits are what we call the tubulin. So as what I said kaina, so microtubules are made of two types of tubulin molecules. One is alpha tubulin and one is beta tubulin. Alpha tubulin at the end and beta tubulin at other end. And cross-section, microtubules contains 13 subunits around the circumference. One end is known as plus end, other end is known as minus end. So the plus end is what we call the alpha tubulin, while the minus end is what we call the beta tubulin. So the microtubules associate protein, or in the abbreviation is MAPS. So recently, some new proteins are identified that associate with the microtubules known as MAPS. Under the electron microscope, MAPS are seen to have globular structure which attach to the side of MT. MAPS increase the stability of microtubules that rigidity influence rate of assembly. So the microtubules associated proteins or MAPS are the one who increase the stability of microtubules in the cell. So they influence the cell to assembly. So they are what we call stabilizing MAPS. There are two types of stabilizing MAPS, the tau and MP2. So the tau are the one who bind side and stabilize microtubules, while the M8P2 or AMP2 bind side stabilize microtubules. So this what we call this stabilizing MAPS. So in this stabilizing MAPS, there are OP18 or stamen that are binds tubulin dimers and destabilize. So AF18 are the one who destabilizing the MAPS. So may ginatawag kita nga katani. This kind of MAPS is destabilizing man. AAA, ATPase that serve microtubules. So in ATPase, the katani are the one who serve the microtubules. Then XKCM or MCA key are the kinesin-related stabilizing plus N. So the XKCM are known as kinesin-related destabilizing plus N. So may ginatawag kita nga CLIP170 is the first microtubules plus N tracking probe. So CLIP170 are the one who link members to grow plus N. So imo ang responsible sa mag-grow ang plus N. So bind sa microtube plus N to reduce catastrophe. So ang kanya nga CLIP170 nga kind of MAPS are the one who reduce the catastrophe in the cell. So maginatawag nga AB1 is another microtubule plus N tracking protein identified. So EB1 are the one who bind the microtubule plus N to reduce catastrophe man. So present the responsibility of CLIP170 at AB1 nga gagareducean the catastrophe sa
microtubules as structural supports and organizers. The distribution of microtubules determines the shape of the cell. Microtubules maintain the internal organization of cells. Microtubules function in axonal transport. Microtubules play a role in axonal growth during embryogenesis. In plants, microtubules help maintain cell shape by influencing formation of the cell wall. They are stiff enough to resist the forces that can bend or compress the fiber. This distribution of microtubules through the cytoplasm of cell determines the shape of a cell or egg. In columnar epithelial cells, the microtubules are aligned along the axis of the cell. It maintains a K in the internal organization of a cell. Microtubules as agent of intercellular motility. They facilitate movement of vesicles between compartments, axonal transport, movement of neurotransmitter across the cell, movement away from the cell body or antegrade and toward the cell body or retrograde. Mediate tracks a variety of motor proteins. Motor proteins traverse the microtubular cytoskeleton. Molecular motor convert energy from ATP into mechanical energy. Molecular motors move unidirectionally along their cytoskeletal tract in a stepwise manner. The three categories of molecular motors are kinesin and dynein move along microtubule tracts, myosin moves along microfilament tracts. Kinesis. Kinesin is a member of superfamily called KLPS or kinesin-like proteins. A kinesin is a tetramer of two identical heavy chains and two identical light chains. Each kinesin includes a pair of global heads or the motor domain connected to a rod-like stalk. Kinesin implies and directs a microtubular motor based on its movement. Kinesins, they move along a single protofilament of a microtubule in a velocity proportional to the ATP concentration. Movement is processive. Motor protein moves along an individual microtubule for a long distance without falling off. KLPS moves cargo toward the cell's plasma membrane. Cytoplasmic dynein. Dynein is responsible for the movement of cilia and flagella. Cytoplasmic dynein is a huge protein with a globular force-generating head. It -N directed microtubular motor. Requires an adapter or dynactin to interact with the membrane-bounded cargo.
I will discuss the microtubule organizing centers or mitochs. Mitochs is specialized structure for the nucleation of microtubules. Second, centrosome is a structure responsible for initiating microtubules in animal cells, and it contains two barrel-shaped centrioles surrounded by parenchyrular material or PCM. Second, centrioles are usually found in pairs. Third, responsible for initiation and organization of microtubular cytoskeleton. And lastly, the microtubules terminate in PCM. Mitochs are also present in all eukaryotic cells that play a role inside of centrosome, which is contains of centrioles and PCM. All mitochs share a common protein component, Y-tubulin, discovered in mid-1980s. It is 0.005% of total cell protein, and while A negative and B negative tubulins are 2.5% of total non-neural cell protein. Fluorescent anti-Y-tubulin antibodies or ABS stain all mitochs like centrosome PCM suggest it is critical component in MT assembly and nucleation. Microinject Y-tubulin AB into living cell blocks MT reassembly after deplomerization by drugs or cold temperatures. Genetically engineered fungi lacking functional G-tubulin gene cannot assemble normal MTS. The mitochs stabilize the microtubules through an associating stabilizing the common proteins. Application mechanism revealed by structure composition studies of PCM at centrosome periphery. Insoluble fibers of PCM are true to serve as attachment sites for ring-shaped structure that have same diameter as MTS, 25 nanometer, and contains white tubulin. Ring-shaped structure found when centrosomes were purified and incubated with gold-labeled anti-white tubulin ABS. Cluster in rings or semicircles at empty-ends, ends embedded in PCM. Isolate similar ring-shaped complex Y TURCS from cell extract nucleic empty assembly and bite through. The centromere consists the PCM. This serves as a platform for proteins that regulate the organelle activities and protein degradation and for spindle assembly. Main function of mitocs: the organization of eukaryotic flagella and cilia and the organization of mitotic and meiotic spindle apparatus, which separate the chromosomes during cell division. Most animal cells have one mitoc during interphase, usually located near the nucleus and generally associated closely with the Golgi apparatus. The mitox is made up of a pair of centrioles in its center, and it is surrounded by pericentriolar material or PCM that is important for microtubule nucleation. Each daughter cells after the process of mitosis is contain primary mitox. During the cell division, the centromere moves to opposite side or end of the cell. Also, the microtubules is help the mitotic spindle. If the mitocs stop working to replicate and the spindle can form, the mitosis will be not complete the process. Basal bodies and other mitocs. Basal body structure where outer microtubules in cilia and flagella are generated. Plant cells like mitocs and their microtubules are organized around the surface of the nucleus. Mitocs can also organize the microtubules that make up the cilia and flagella. It stabilizes and gives a direction to the microtubules with the centrosome.
Dynamic properties of microtubules. There are four distinct arrays of microtubules in dividing plant cells, widely distributed throughout the cortex, making a single transverse band in the form of mitotic spindle, as a pharmaplast assessing in the formation of cell wall of daughter cells. Microtubules vary markedly in stability even though similar morphological spindle, cytoskeleton level, mature neuron MTS less level, cilia, flagella very stable labilities allow to respond to stimuli. Cilia flagella microtubules are stabilized by AMP attachment and by enzymatic modification. Example is acetylation of specific amino acid residue with tubulin subunits. Label microtubules in living cells can be disabled without disrupting other cell structure via a number of treatments.
The newly formed microtubules branch an angle of pre-existing microtubules. The changes in spatial organization microtubules are a combination of two mechanisms. This mechanism helps the microtubules rearrange and disarrange. Rearrange of existing microtubules, disassembly of existing microtubules, and reassembly of new one in different location. So, the special organization are the one who organize and who arrange the existing microtubules. So, there are treatments that cause microtubules disassembly. Usually, it interferes with non-covalent bands to hold them together. So, there are some treatments that will cause microtubules disassembly. So, these are the cold temperatures, hydrostatic pressure, elevate calcium concentration, variety of chemicals are often used as chemotherapy or CO, vinblastine, vincristine, and O, pudifilitoxin. Those are the micro the causes of why the microtubules disassembly. So, some treatments or Taxol disrupt the microtubules' dynamic activity and act by doing the opposite, inhibit disassembly. So, the Taxol binds microtubules polymer and thus prevents disassembly. Cell cannot build new microtubule structure. So, if the microtubules structure will going to disassembly, the cell cannot produce or cannot build a new structure of microtubules. So the cytoskeleton microtubule lability reflects fact they are polymers formed by non-covalent association of dimers, subject to the repolymerization as need to change. So, the dramatic changes in microtubule spatial organization may achieve by combination of two separate mechanisms. These mechanisms are the rearrangement of existing microtubules and the disassembly of existing microtubules and reassembly of new ones in different cell region. So, the study of microtubules dynamics in vitro suggests that cytoskeleton can be rapidly remodeled and respond to stimuli. So, in early studies established that GTP binding to beta subunits requires for microtubules assembly. GTP hydrolysis not needed for binding, but it is hydrolyzed soon after dimer attached to empty and GTP stays bound. After the dimer is released from microtubules during the disassembly and enters soluble pool, GTP is replaced by GTP, thus recharging dimers so that it can add to polymer again. A GTP molecule is bound to an alpha-tubulin subunit but is not exchangeable and it is not hydrolyzed after subunits. Assembly is not energetically inexpensive since it includes GTP hydrolysis, but it does allow the cell to control assembly and diassembly independently. So the GTP hydrolysis are the one who allow the cell to assembly and disassembly independently. So they are the one who control the microtubules assembly and disassembly. So a dimer being added to empty or microtubule has bound GTP. Dimer being released from microtubule has bound to GDP. So the GDP and GTP dimers have different conformation and participate in different reaction. The ends of growing and shrinking and microtubules have different structures. So the GDP and GTP dimers has a different conformation or they have different structure when it comes in the cell. So they also separate different kind of reaction. That's why they are differ from each other. So the shrinking of microtubules has different structure. So when the microtubules shrinking in the cell, so they has different structure will going to form. So the above facts lead to following model. So there are different kinds of model when it comes in microtubules. So when a microtubules growing, the plus n is represented as the open sheet to which GTP dimers are added. If the microtubules grow at plus m, it will represent the GTP dimers. During rapid growth periods, tubulin dimers are added faster than GTP can be hydrolyzed. The resultant cap of GTP dimers on microtubules at protofilament ends is true to favor the addition of more subunits and hence microtubules growth. However, microtubules with open ends, though, to undergo spontaneous reaction leading to tube closure. So if the microtubules ends undergo spontaneously, the reaction will going to close the tube in the cell. So tube closure is accompanied by hydrolysis of bound GTP, changing tubulin dimer conformation resultant mechanical strain belies MTS. Strain is released as protofilaments uncurl from tubulin catastrophically depolymerize. This assembly can occur remarkably fast, especially in vivo, which allows very rapid microtubule cytoskeleton disassembly. The study of microtubule dynamics in vivo, dynamic character of microtubule cytoskeleton inside cell is best revealed by microinjecting label tubulin into non-dividing culture cell. So in injecting label tubulin into non-dividing culture cell, it labels the subunits rapidly incorporated into pre-existing cytoskeleton microtubules, even in absence of any obvious morphological change. Watch cell with fluorescent label microtubules over time. Some microtubules grow, others shrink, other is dynamic. Both grow and shrinkage in vivo occur predominantly at plus end of polymer, the end of location opposite the centrosome or the other MTOC. Single microtubules switch randomly and unpredictable between growing and shrinking dynamic instability. Microtubules shrink faster than they go, so in a matter of minutes, microtubules disappear are replaced by new microtubules that grow out from centrosome. The underlying of microtubules dynamic, insight into factors that influence microtubules assembly and disassembly, came from study in vitro. GTP is required for microtubules assembly, like hydrolysis of GTP leads to represent a bond GTP by new GTP to recharge the tubulin dimer. Interaction between microtubules and actin filaments. Microtubules and actin filaments as function together during locomotion of cell and cell division. So the microfilaments and actin filament has the function during cell locomotion and cell division. They are the one who help the cell to move and to regulate the cell division in our body. In general, microtubules direct wear and when acting assembly or generates contractile forces. Microtubules influence the actin cytoskeleton. The two cytoskeletal system can be found together by linker proteins that attach microtubules to actin filament.
My topic is all about flagella and cilia. Flagella are much longer than cilia and are usually present singly in pairs. A single flagellum may propel the cell with a whip-like motion. A pair of flagella may move in synchronized manner to pull the organism through the water, in a way similar to the breaststroke of human swimmer. So, si flagella, mas mahaba siya kaysa kay cilia kasi mas responsible siya sa movement ng mga cell. Pag-whip motion siya kasi yung pag-move, yung motion niya kasi parang pa-letter S. Then, ang pair ng flagella ay nabumove siya kasabay sa paghatak ng organism sa tubig. So, similar siya sa breaststroke mga swimmer. Cilia tends to cover the surface area of a cell. Both cilia and flagella bend as a microtubules like to pass to one another. The arrangement of cilia permits their coordinated movement in response to signal from the cytoplasm. A small ciliate may have hundreds of individual cilia all beating in a coordinated manner. So parehong si cilia at si flagella, nagbe-bend sila sa microtubules in one another. Flagella and cilia, this extension from the cell surface aids cell movement. They also help to move substances around the cell and direct the flow of substances along the tract. The cilia and the flagella are formed from specialized groupings of microtubules called basal bodies. So yung basal bodies na to is protein structure siya na makikita sa base eukaryotic endolipidium. So if the protrusions are short, and numerous, it is a cilia. So if they are longer and less numerous, they are flagella. Cilia and flagella have a coin composed of microtubules that are connected to the plasma membrane and arranged in is what is known as 9+2 pattern. So si cilia, si flagella, at meron silang cord na nagko-compose ng microtubules, tas nagko-connect ito sa plasma membrane at nag-arrange ito kung ano yung mga pattern. The pattern is so named because it consists of a ring of 9 microtubules per set, droplets that encircles 2 singular microtubules. So yung mga pattern na ito may mga pangalan kasi ito ay yung parang siyang ring na may 9 na pair ng microtubules. These microtubules bonded in a 9+2 arrangement is called axonomy. The base of cilia and flagella is connected to the cell. Modified contriole structure called basal bodies. So yung base ng cilia at ng flagella ito ay nagko-connect sa cell para ma-modified yung structure nito na tinatawag nating basal bodies. Movement is produced when 9 pairs microtubule sets of the axonomy slides against one another, causing cilia and flagella is bent. The motion protein dynein is responsible for generating the force required for movement. So yung protein ng dynein ay mas responsible siya para ma-generate yung force na nire-require sa paggalaw ng dynein. Central empties enclosed by projections forming central sheath. Sheath connected to doublet A empties by radial spokes. Double connected by inter-doublet bridge made of elastic protein hexine. So yung sheath is nagko-connect siya sa doublet AT empties ng radial spoke. Yung double connected ay meron siyang bridge na gawa sa elastic protein hexine. Pairs of arms, inner and outer, project from AMTs in clockwise direction. So pairs of arms is lang siya, clockwise. Yung direction niya is clockwise lang. Radial spokes, typically in groups by 3 with major repeat of 96 nanometer. Inner and outer dynein arms target along a empty's length. Outer arm space every 24 nanometer, inner arms arranged to match unequal spacing of radial spokes. The mechanism of ciliary and flagellar commotion sliding filaments model suggested mechanism of ciliary and flagellar movement was sliding of adjustment empty doublet relative to one another. So suggested sila ng mechanism ng ciliary and ng flagellar para sila ay makagalaw. In model, dynein arms act so swinging cross bridging that generate forces needed for ciliary flagellar movement. Dynein arms projecting from one doublet walk along adjustment double wall. So kailangan ng ciliary at flagellar yung paggalaw ng dynein arms para ma-projecting sila galing sa doublet ng adjustment ng wall. Sequence of event in ciliary flagellar sliding motion. First, it is dynein arms anchor on a doublet's AMT attached to binding sites on BMT of adjustment doublet. Second, dynein molecules undergo conformation changes causes AMT doublet to over slightly toward basal end attachment BMT of doublet. Third is dynein then releases B tubules of adjacent doublets. Last, dynein are reattached to adjacent doublets BMT closer to its base, so another circle. Cilia and flagella are motile structure. Cilia and flagella contains a highly ordered coin structure called an axonomy. So yung cilia, si flagella they have a core structure na tinatawag nating na axonomy. The axonomy is composed of outer doublet microtubes surrounding of a pair of central microtubules. Radial spokes of complex of several polypeptides link each outer doublets to the center of the axonomy. Dynein are bound to each doublet and extend their motor domains towards the adjacent outer. Dynein slides the outer doublet past each other. Kinesis particular in flagella assembly by transporting axonomy proteins to the distal. So yung kinesis ay kasama siya ay nagpa-participate siya sa flagella para ma-transport yung axonomy sa proteins. So ito yung structure ng cilium or flagellum.